let's talk a little bit about the hormone epinephrine, or as we like to think of, adrenaline, right? Hormones act as signal molecules. And when epinephrine is released into your blood, it causes different cells to behave differently. So, for example, cells of the eye, pupils, for example, dilate. Tear formation is inhibited. That makes sense, right? Because if I'm about to get into a fight and my adrenaline is rushing, I don't want to start crying and lose the ability to see my target. I want to be able to get as much light as I can into my eye so I can see with better precision. Therefore, I'm going to dilate my pupils and inhibit my tears in response to adrenaline. It's another type of cell that's affected, right? Those cells that produce saliva, all right, those glands. Salivation is decreased in response to adrenaline. Glucose is released from the liver in response to adrenaline. Now, why do you think that is? Why would glucose levels in the blood need to be increased? That's right, because as we learned, glucose is a source of energy. And again, if I'm about to get into a fight, I better make sure that my muscles have enough energy to support the strenuous movement of me running away. All right? Now, what form is glucose stored in the liver? That's right, the storage form of glucose is glycogen. So what's going to happen in response to adrenaline is liver cells are going to produce enzymes in order to convert glycogen back to glucose. And once glucose is produced, that glucose is taken to the blood, and in the blood it's taken everywhere else that requires energy. Other examples of cellular responses in relation to adrenaline: constriction of blood vessels to increase blood delivery or increase the rate of blood delivery. Sweating to regulate body temperature due to increased metabolism. We're going to relax the airways. So you can see here cells in charge of carrying blood, right? Blood vessels are constricted in response to adrenaline, but a different type of cell relaxes in response to adrenaline, and that's to increase oxygen intake. Now, why is oxygen important in response to adrenaline? That's right, oxygen serves as the final electron acceptor in the ETC, and ultimately the energy released in that electron transport is going to be stored in ATP, which I certainly need in response to a stressful situation. Lastly, we're going to speed up the heart rate in response to adrenaline. Heart muscle cells are going to contract and relax more rapidly. And by contracting and relaxing more rapidly, by increasing the heart rate, we're going to increase blood delivery. So the reason I've shown you this is it gives you an example. One individual signal molecule, like adrenaline, can cause different cells to have different responses. And how can cells change these responses? It's through signal transduction. So I'm going to give you two specific examples today. I'm going to give you an example of how adrenaline stimulates the liver to release glucose, and I'm going to give you an example of how adrenaline speeds up the heart rate. First, we have to understand where adrenaline comes from. Okay? Adrenaline is secreted by an adrenal gland. It's a hormone that is carried through the blood. So now recall that this epinephrine, adrenaline, is secreted in response to what we call the fight or flight mechanism. Right? When we're put in a stressful situation and we require an increase in blood flow, when we require our muscles to acquire more energy, it's the epinephrine that's released from those adrenal glands and carried through blood vessels to different parts of the body. So now let's look at the liver. Let's look at, in this example, how the binding of the hormone epinephrine to beta-adrenergic receptors on the surface of liver cells increases the production of glucose and the release of glucose from those liver cells. Now, what I will tell you is this. When epinephrine binds to these receptors on liver cells, this cellular response is mediated by the cyclic AMP pathway. So before I show you how cyclic AMP ends up causing a cellular response, I'd like you to take what you've learned and do that for me. So what I'm going to do is I'm going to ask you to place these series of events in order. Starting from number 1 would be the binding of the signal. In this case, binding of the epinephrine. And then the last item on the list should be the cellular response. So take a moment, try to put them in order using the numbers here. What you're going to want to do here is press the pause button on the virtual lecture so you can then look at these choices. And then press play again once you've ordered them, and a question will pop up. All right, let's look at the pathway now that you've gotten it correct. Now, as I told you in the previous slide, epinephrine binds to a specific type of receptor on the surface of cells, liver cells, called beta-adrenergic receptors. Now, this is a G protein coupled receptor. It's located on the surface of liver cells. So now let's watch what happens once a receptor is bound. So let's do the step through here. Here we can see the release of the hormone from the adrenal gland through the blood vessels. Those particular hormones will arrive at the liver, where epinephrine, as we'll see here, is going to bind these beta-adrenergic receptors. Now, what's going to happen to these beta-adrenergic receptors once the signal is bound? That's right. These receptors are going to change shape. They're going to undergo a conformational change that's going to be transmitted from the extracellular side all the way to the cytosolic side. Okay? So now once that happens, we can see that the change in receptor shape is ultimately going to activate the G protein. Now tell me, how do we activate this G protein? Before I show you the animation, how is this G protein activated? That's right, it's activated by nucleotide exchange. In this case, what's going to happen is once that G protein is bound to activated beta-adrenergic receptor, GDP is going to fall off, and that GDP is going to be replaced by GTP, which puts that G protein into the on state. So here we see the binding of G protein to the G protein coupled adrenergic receptor is going to result in a change in the G protein. When this change occurs, GDP is removed, and now GTP is added. Now, upon binding the GTP, the G protein is going to dissociate, the alpha subunit is going to be freed, and now once that's freed, that G protein is going to activate adenylate or adenylyl cyclase. Now, what is the function of this enzyme, adenylyl cyclase? That's right, adenylyl cyclase catalyzes the chemical reaction of converting ATP to cyclic AMP. So what happens now is adenylyl cyclase is going to be turned on upon binding to the G protein. You can see here through these series of events, we're going to have an increase in cyclic AMP levels in the cytosol. Now, once cyclic AMP levels are raised in the cytosol, they can now carry the message. So if we look, here's the adrenaline or the epinephrine signal molecule. It stays outside the cell, but now through a series of steps, we now have a small chemical signal located inside the cell called cyclic AMP. Now, once cyclic AMP is activated, as we talked about, what is cyclic AMP going to go do next? That's right, as we talked about, cyclic AMP activates protein kinase A. So here we have that small molecule. What's happening here is you have your regulatory subunit of protein kinase A. Here's your catalytic subunit. Now, which of the two is responsible for the addition of phosphates to other proteins? Is it the regulatory subunit or is it the catalytic subunit? That's right. The catalytic subunit has the kinase activity. Therefore, what's the function of the regulatory subunit? As we talked about previously in this lecture, a function of the regulatory subunit is to turn off the catalytic subunit. So when we have this complex, protein kinase A is inactive. And the way in which we get this to dissociate from the catalytic subunit is the binding of cyclic AMP into these specific binding pockets. So now what happens is as cyclic AMP binds the regulatory subunit, the complex falls apart, and now our catalytic subunits are available. In this case, you can see here is the nuclear envelope pictured in this animation. Here are our nuclear pores, which will allow the catalytic subunit to enter the nucleus. Now we can see once in the nucleus, there's a phosphorylation event. We have active protein kinase A adding a phosphate to this transcription activator. Don't worry about the name of this. Okay, just want you to see the cellular response. You're not responsible for this part of the pathway. So you should be able to describe to me everything that's happened prior to this, but once we get to this point, we're just going to describe this as pKa is going to phosphorylate a protein that increases mRNA transcription. Now, if I take a step back, recall the effects of epinephrine on a liver cell is to increase the glucose release into the blood. And if you recall, glucose is stored as glycogen. So what we need to do as liver cells in response to adrenaline is we need to increase the amount of enzyme required to break glycogen down to glucose. And so this target gene that's shown here, and let me try to clear up some of this clutter. This target gene that's shown here is a gene that produces the enzyme required for glycogen breakdown. So as you can imagine, we need more glycogen breakdown in order to respond to adrenaline. What's one way in which we can increase glycogen breakdown? How about increasing the amount of enzyme available for that breakdown? And how do you increase the amount of a protein in the cytosol? You can increase the amount of RNA for that protein. And how do you increase the amount of RNA for a protein? You can increase the rate of transcription. So that's what we're going to see here. So now we have this activated transcription factor called CREB. It's going to recruit a couple other accessory proteins. Again, don't worry about the names of these. But as a result, we're going to increase transcription. We're going to produce the RNA for this particular enzyme. Now what's going to happen is that RNA is going to travel out to the cytosol, where we then undergo translation to make the enzyme required for glycogen breakdown. And so now as a result of epinephrine binding to its receptor, we are going to increase glucose production. So what you saw there is the role of cyclic AMP in the process. Cyclic AMP took the signal, received the information, and then caused a cellular response by ultimately increasing glucose levels. Okay, so now that we've looked at how cyclic AMP can react to the binding of adrenaline in specific cells, in that previous case, liver cells, let's look to see how calcium actually works. And in this case, we're going to see the effect of adrenaline on heart muscle cells. Now, what do we have to do to the heart rate in response to adrenaline release? What's going to happen to the heart rate in response? That's right, as we talked about previously, we're going to increase heart rate in response to the fight or flight. So when we're under stress, the heart rate is going to increase, which is going to increase the delivery of blood to the rest of our cells. Now, before I show you how calcium can mediate such an event, I want you to put the following events in order. Starting with signal, which is epinephrine binding the beta-adrenergic receptor on the surface of heart cells, and then ultimately to the cellular response, which is an increase in heart rate. So press pause now on your video, on your virtual lecture. Put these in order. When you're done putting them in order, a question's going to pop up, and let's continue with the lecture. This is exactly what we talked about before. In this case, here's my epinephrine. Right, that's my signal molecule. Here is my G protein coupled receptor, a beta-adrenergic receptor. Activation of the receptor, right, causes a G protein to be activated. Now, instead of working through adenylyl cyclase, in this particular case, the G protein is going to work through a intermediate molecule called an effector molecule. Once that effector is activated, it's going to cause a release of IP3. IP3 is going to open those calcium channels on the surface of the ER. Opening up those calcium channels, it's going to increase cytosolic calcium concentrations, and those calcium ions are going to activate a protein called troponin. And because we've activated that troponin, what's going to happen is muscle contraction is carried out. So we can see that in this example, when calcium is elevated in the cytosol of a heart cell, this increases the contractions. How does this happen? When calcium binds to these troponin molecules, which are part of these actin filaments, once calcium is bound, that's going to cause the muscle cell to contract, which causes then the pumping of blood out of the heart. So again, we can see how adrenaline ultimately leads to the shortening of a muscle cell, in this case a shortening of the actin filament through the binding of calcium. Now what's interesting is calcium in the cytosol is going to cause that muscle to stay contracted. So just as we have to contract the heart muscle cell, we also have to relax the heart muscle cell, because the contraction is going to pump the blood. The relaxation is going to allow the heart to fill back up with blood. So how can then we get the calcium back out of the cytosol into the ER against equilibrium? What kind of transport are we going to require? That's right, we're going to require active transport, as we discussed earlier in this lecture. So we're going to require these carrier proteins to bind calcium. We're going to require a phosphate to be added in order for that calcium to be taken back into the ER against the gradient. So how in the world can we regulate this calcium pump in response to adrenaline? Here it is again, our favorite, cyclic AMP can help. So when epinephrine binds to the receptor on the surface through another series of events, cyclic AMP levels increase, which results in the activation of pKa. pKa phosphorylates a protein, don't worry about the name of it, called phospholambin. Phospholambin actually increases the rate of active transport to clear the cytosol of calcium, to put all those calcium ions in high concentration, to return the cytosol back to low concentration. And it's this recreation of the gradient that relaxes the heart muscle cells. So what you can see is the heart responds to adrenaline in two ways, right? The heart responds to adrenaline by causing calcium to spill into the cytosol in order to cause muscle contraction. It also activates the cyclic AMP pathway in order to activate calcium pumps in order to clear that calcium. Now, one of the interesting things, or one of the interesting side effects of caffeine is that it increases your heart rate. Your heart actually contracts a little bit more vigorously in the presence of calcium. In the presence of caffeine. Now, why is that? Well, what's interesting is calcium prevents the breakdown of cyclic AMP. So remember, we have an enzyme called phosphodiesterase that hydrolyzes the cyclic AMP here to go back to the AMP form. Now, if cyclic AMP can't be broken down, what's that going to do to phospholambin and even the troponin? That's right, if you cannot break down cyclic AMP, it's going to increase the contraction by troponin. It's going to increase the rate of relaxation due to the actions of the phospholambin. So what that's ultimately going to do is increase your heart rate artificially. So I think that's interesting. Right? This enzyme, phosphodiesterase, is inhibited by that molecule, caffeine. So let's summarize now. How does adrenaline work? Adrenaline binds to beta-adrenergic receptors on the surface of heart muscle cells. When those heart muscle cells are bound, two pathways are stimulated. First, we have the cyclic AMP pathway, which increases the rate of muscle relaxation and even muscle contraction. The second pathway that's activated, which is the calcium pathway, also leads to an increase in the force of that muscle contraction.